Hey everyone, I'm Robin and I'm the lead author on this paper. I wanted to give a quick overview of the key points from our new paper, From Defaults to Databases, Parameter and Database Choice Dramatically Impact the Performance of Metagenomic Taxonomic Classification Tools, which is out now in Microbial Genomics. I want to start by giving a really quick overview of the motivation behind this study. As you may be thinking that there are a lot of studies that compare metagenomic taxonomic classifiers out there already, and you'd certainly be right, but what these don't do is compare the impact of different parameters and reference databases on the classifications obtained. One of the first steps in the analysis of metagenomes is often the taxonomic classification of reads, and there are two tools that have been the most widely used for this to date, Metaflan3 and Kraken-2. We had initially assumed that the performance of these classifiers would be somewhat comparable, and we started out with a quick test of which tool and parameters we should use to classify some samples. And I'll just demonstrate the kind of things that we found using a very pared down example of part of Figure 1. If we look at a selection of human-associated metagenome samples that also have paired 16S rRNA gene sequencing, then the 16S amplicon sequencing tells us that we have a median of 117 species, or ASVs, per sample. If we classify the metagenome samples using Metaflan3, then this would tell us that we have a median of 64 species in those samples. But classifying those same samples with Kraken-2 and a very complete reference database gives us between 22,188 species with the default settings, or 568 species with the most stringent settings. And this issue actually gets even worse if we look at environmental metagenome samples that also have 16S sequencing results. So here the 16S sequencing identifies a median of 426 species, Metaflan3 a median of 0 species, and Kraken-2 between 33,135 species with the default settings or 115 with the most stringent. So here we can clearly see how using each of these two tools, Metaflan3 and Kraken-2, could create extremely disparate results. So how do we know which of these is correct or the closest to correct? Well, we took approximately 400 previously made simulated metagenome samples, and we used both Kraken-2 and Metaflan3 to classify them taxonomically. For both tools, we used a range of different parameters within them, which I'll go into in a little more detail as we get started. And for Kraken-2, we also used some different databases, all of which are available on Dropbox. We tested out almost all of the tool parameter database combinations that we could, and we took a really deep dive into Kraken-2's confidence threshold, and this gave us above 60,000 taxonomic profiles for us to analyze. So we were hoping that in doing this, we could answer this question of which tool parameter database combination is best. And in order to explain the parameters and databases that we used, I need to explain a little bit about the tools first. As I mentioned, one of the first steps for any metagenomic taxonomic classifier is usually the taxonomic classification of reads in our samples by comparison to a database of previously taxonomically classified genomes. And the two main approaches that are used to do this are a marker gene approach or a KEMA-based approach. The first approach that's often taken is to take our database of genomes and to either only take marker genes, like the 16S ribosomal RNA gene, or to take genomes from similar taxa and identify marker genes that are unique to these taxa that we can then search for in our sequenced reads. This obviously means that we will only be classifying the reads in our samples that contain those markers, but we can typically be fairly confident in the taxa that we identify in this way. So Metaflan3 uses a database called Chocoflan3, which is comprised of about 17,000 species containing almost 100,000 genomes. Using this curated database of genomes, they've identified 1.1 million taxonomic marker genes, most of which come from bacteria, but others are also from archaea, eukaryotes, and viruses. So Metaflan will map the reads in the samples to this database of marker genes, and then it will use this to estimate the number of reads that would be contributed by each taxon from the whole sample. The other approach that's often taken is a KEMA-based approach. So with KEMA-based tools, basically we take our database of genomes, and for each of the genomes, we calculate all subsequences of length K, which are then known as KEMAs. And these KEMAs are what's used to build our database. A tool like Kraken, which is currently on its second version, will then take a query sequence and will divide this up into KEMAs, which it will map against the previously made database. So in the example here, of the 27 total KEMAs in the query sequence, 11 are unclassified, 10 of these came as mapped to the taxon in blue, 4 to the orange taxon, 1 to the red taxon, and 1 to the purple taxon. And here the purple taxon is the ancestor of all mapped reads, and the blue taxon is the ancestor of the orange taxon. Now, based on these mappings, we can work out how confident we are that our sequence belongs to one of these taxa. So in this case, the sequence would be classified to the leaf node, or the lowest rank possible, and our confidence that the sequence is this taxon is 4 out of 27 kmers, or 0.15. Our confidence that it's the blue taxon is 10 plus 4, or 14 out of 27, or 0.52, and our confidence that the sequence belongs to the purple taxon would be the sum of all leaves here, so 0.59. And Kraken allows us to set a confidence threshold. So in this example, if we have no confidence threshold set, then this is 0 by default. So we'd say that the sequence is the orange taxon. If we set it to say 0.2, then we'd say that the sequence belongs to the blue taxon, and so on. So because with Kraken 2, it's really easy to make databases with just about any group of genomes we have, we started to test a range of databases on our simulated samples with Kraken 2. The top database here is the largest, and it contains everything in the NCBI RefSeq database. So that's all genomes. And this also includes those that aren't complete. On the left, we have the database size, and this one's almost 1.2 terabytes. In the middle, we have the number of taxa, and this contains about 110,000 taxa. And on the right, this is the proportion of the taxa from our simulated samples that are covered by this database. Each point here is an individual sample with the box plot showing the median and interquartile range, and you can see that it covers about 99% of them. I won't go through all of the rest of them. I'll just highlight another 2. So next, we have the Chocoflan database. And this is the closest equivalent to running Metaflan 3. It's only 73 gigabytes. It contains all of the genomes from the Chocoflan 3 database, and it covers about 60% of the taxa in our simulated samples. And then finally, this database on the bottom is the smallest database. It's linked from the Kraken 2 website, the mini Kraken database, and it also contains the lowest proportion of taxa from the simulated samples. So then we started to look at the effect of changing some of the parameters within Kraken 2. And here we did look at some other parameters as well, but they didn't make too much of a difference. So we focused on the confidence threshold. And here I'm focusing only on the RefSeq complete database because it covered the largest proportion of taxa. So first of all, as we increase the confidence threshold on the x-axis, we see that, unsurprisingly, the proportion of reads within samples that could be classified decreases. And here the grey lines are each individual sample, and the red line and shaded area is the median and upper and lower quartiles. If we then look at the dissimilarity in the number of species that are identified, so here with a confidence threshold of 0, there are thousands more species identified by Kraken than in the known composition. And the difference between the two is minimized at confidence threshold of 0.65. And then by a confidence threshold of 1, we just have in the region of 10 fewer species identified by Kraken than in the known composition. And if we finally look at a metric of beta diversity, the L1 distance, then we see that the distance between the Kraken classifications and the known composition decreases ever so slightly initially, and it's minimized at a confidence threshold of 0.15, and then it increases with increasing confidence threshold. And then here I'm just showing some common performance metrics, the precision, recall, and F1 score. And each of these say mean because they were the mean of the metrics calculated on the taxa classified as well as the reads classified. So we see that as the confidence threshold, again on the x-axis, increases, so does the precision, but it peaks at a confidence threshold of about 0.8 and then decreases. The recall decreases as confidence threshold increases, and then the F1 score is maximized at a confidence threshold of 0.6. So the optimal confidence threshold to use depends on which metric we focus on, and it essentially comes down to whether high precision or high recall is more important to us, or the question that we're wanting to answer with our data, and really whether we're more concerned with false positives or false negatives. If we now combine looking at the confidence threshold with looking at different databases, here I'm showing a slightly different graph with the proportion of reads classified, dissimilarity in number of species identified, a mean F1 score making the different panels from left to right. And then we have our different databases on the y-axis and confidence thresholds again along the x-axis. Here we have increasing database size from bottom to top, and we basically see that while the optimal confidence threshold to choose does change with database size, the overall trends for each are similar, with the proportion of reads classified decreasing with increasing confidence threshold until a point. And then the dissimilarity in the number of species classified generally decreasing with increasing confidence threshold until a point, and then increasing again. And the mean F1 score increasing with increasing confidence threshold, again up until a certain point at which it starts to decrease again. And generally, the smaller the database, the lower the confidence threshold at which the most optimal values are obtained for the dissimilarity in number of species identified, as well as the mean F1 score. But the best scores for each metric are always with the NCBI RefSeq complete database on the top row. And I do just want to mention that we did test out some different parameters within MetaFlan3, but we found that the default already performed well. We tested out some different Bowtie2 read mapping to the marker gene database options, some different statistical methods for calculating the number of reads that were classified, and some different options for looking at only the actual reads that were mapped to the marker gene database. So they were the Human3 options. And you can see that for most of the options, aside from the Human3 ones, while the estimated proportion of reads classified did vary, the mean F1 scores weren't very different. And the dissimilarity in number of species identified was generally reasonably similar. These aren't changing by, an order of magnitude in the thousands as they were for Kraken. And we therefore thought it was best to just stick with the default here, as the tool creators have clearly done a decent job of choosing the default parameters. So now if we finally compare Kraken 2 and MetaFlan3, we chose to look at the best performing NCBI RefSeq complete database, as well as the ChockerFlan3 equivalent database for Kraken. And for each, we just looked at confidence thresholds of 0, 0.5 and 1, as well as those where the mean F1 score was maximized or the L1 distance was minimized. And then for MetaFlan3, we just went with the default and the slightly less sensitive, and also the sensitive Bowtie2 option. So this was just slightly less stringent than the default very sensitive option for mapping of the reads in samples to the marker gene database. So here, when we look in all of those simulated samples, so each of the points, we see that while there is some variation in which tool performs the best, generally Kraken with the RefSeq complete database can have a more optimal value than MetaFlan3. Although I think it's really important to point out that if we just use the default of no confidence threshold with this, then we would identify over 4,000 too many species, and the F1 score would only be 0.385, while for the optimal value, we identify only 4 too many species, and the F1 score is 0.684. For the ChockerFlan3 database, we always identify too many species, but the optimal F1 score of 0.55 is still higher than for either MetaFlan option. And then for MetaFlan, with either Bowtie2 option, we identify too few species, but the F1 scores of 0.542 or 0.513 are higher than most of the non-optimized Kraken 2 F1 scores. Okay, so just to summarize briefly, Kraken can work really well, but it definitely won't if you just use the default parameters or databases, and the optimal parameters to use will depend on the database you use, as well as what the question you're trying to answer is. MetaFlan3 is really quick to download and run. The database is only 2.8 gigabytes. And if you're doing microbiome research for a really well-characterized environment, then it probably will give you a nice quick overview of some of the taxa that are in your samples, but you just need to be aware of the limitations and that you will likely be missing a lot of taxa. And then finally, we actually have a small flowchart suggesting how you should decide on which taxonomic classification method best fits for your analysis. I'll highlight again that Kraken-2 is capable of producing more accurate classifications than Metaflan-3, so we do suggest using it where resources allow. But if your samples are from well-characterized communities, and it doesn't matter to you whether you have classifications for each read individually, the Metaflan-3 works really well right out of the box. If you choose to use Kraken-2, we suggest using the largest database that your resources allow. And I will just note that we made all of our databases available to download, and we also have instructions for using the Amazon Web Services instance that already has everything pre-installed in it. And that's all on our GitHub. We also have instructions for creating databases, scripts for testing the performance of those databases, and we will in due time have links to updated databases. So after deciding on a database to use, we recommend running a test with a subset of your samples and a variety of confidence thresholds in order to identify the highest confidence threshold that still has an acceptable proportion of reads classified. And if in doubt, if you're more concerned with high precision, increase the confidence threshold. And if you're more concerned with high recall, decrease the confidence threshold. So thanks very much if you've listened to this overview of our paper. I hope it was useful and that you've enjoyed it. This was just a small selection of the results in the paper, so please do check out the full paper that's out now in Microbial Genomics, and don't hesitate to reach out with any questions. Thank you.